Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last couple of classes, we have been talking about gene expression analysis followed by WGCNA or weighted gene co-expression network analysis. We talked about the theory of WGCNA and then we started working on a hands-on problem on a data set in cancer. So, we will be continuing that today. So, I will just briefly remind you the data set that we worked with. So, this was a gastric cancer data set, diffuse gastric cancer, and then there are also normal samples. And so, we will be continuing that, and we did some of the steps and we will be following that up today in this class and the next class to complete the the hands-on. So, these are the topics that we will be talking about today. The first is soft threshold determination and the network construction. This is the heart of the WGCNA analysis, and the idea of this soft threshold instead of a hard threshold is to identify the threshold where the network becomes a scale-free network. Okay. So, this is something you will see in this class. The term the scale-free network will appear very frequently. So, what is scale-free network? So, we will have that discussion first and then we will go into the hands-on. So, what is a scale-free network? So, a scale-free network is a network where the degree distribution in the network follows power law. Okay. So, I will explain this and elaborate this a bit more. So, let us say K is the number of connections that a node has with other nodes. Okay. So, that is the degree, that is what we define as degree in graph theory. So, if K is the number of connections or the degree, then and if Pk is the probability distribution of nodes with K degree, then Pk should follow K to the power minus gamma. Okay. So, that is the power law distribution, right. So, why are we interested in this scale-free network? Because it has specific property. So, what is this property? That we have a lot of vertices that have degree greater than the average, okay, and we can find hub genes in this network. So, why are we interested in hub genes and what are the hub genes? So, hub genes are kind of like the center of network, if you will. So, they have a lot of connections with other genes. Okay. So, these hub genes are of interest to us because these are likely to control the behavior or regulate the behavior of other genes that it is connected to, right. So, in many cases these hub genes could be transcription factors, right. So, these genes, these transcription factors can regulate activity of many other genes. So, that is why finding hub genes is actually important for this WGCNA analysis. Now, when you find hub genes, these are likely to be the driver genes in a process that we are interested in, that we are studying. In our case, we are studying this diffuse type gastric cancer. So, in that case, finding a hub gene could tell us, okay, these are the transcription factors or the genes that are perhaps important for development of gastric cancer. So, in that case, these could be interesting targets. So, if this is clear, so we use this soft threshold to actually identify a value where we get a scale-free network, so that we can then follow that up with network construction and then we can find the hub genes. Okay. So, that is the idea for soft threshold determination and that is the motivation. So, now moving on to the workflow, WGCNA workflow. This is something we have discussed already. So, the first step is data loading, then we have normalization and then we have gene filtering. So, these three topics we talked about in the last couple of classes. The data loading, I have shown you where the data is, what kind of data it is, how many samples are there. So, there are many samples in the data set that we took, but we have been working with 9 cancer samples and 9 normal samples just at the interest of time, because we want to complete this in the class. You can of course work with bigger data sets, but they will take more time to learn to run in your system. So, that is something up to you to practice and test. Okay, so the first step is the data loading. We looked at the data and we have loaded the data. This is gene expression data for cancer versus normal, and then we followed that up with normalization. So, normalization, we used the DSEC2 package, and there is a specific method of normalization that we used. This method is called median of ratios method, and this is implemented in the DSEC2 package. There are of course many other methods of normalization, and you can choose other methods that you think would be more suitable in your data, but of course the method that we use is actually quite standard method. Then we did some gene filtering. So, the idea behind gene filtering is to identify the most interesting genes. Okay. So, we can do the WGCNA with all genes that we have, but then we might end up identifying a lot of network, a lot of modules that are probably not very interesting to us. The genes that show difference in expression between cancer versus normal, those are likely to be the most interesting genes. So, we actually used some crude form of gene filtering, right, based on the variance of the gene. Okay. But of course, we can do more sophisticated analysis of gene filtering, where we can do a differential expression analysis between cancer versus normal and only choose expression values of those genes to actually do the WGCNA analysis. Okay. So, that is also possible, but we will not do that here. We have discussed differential gene expression analysis in a separate class, separate course. So, of course, you can combine these and you can come up with a much more sophisticated gene filtering method. Now, we took top 10% genes, the genes that show the highest variance between cancer and normal samples, right, across all samples, and then we will be working with those 10% genes. Now, with these genes, now what we will do in this class, we will be determining the soft threshold. We will see in a moment how we actually determine the soft threshold, and then we will do the network generation. Okay. So, once we generate the network, we will then do the we will store the network data, right, so that we can visualize with other tools, maybe do some network analysis as well. Then followed by that, we will do the identification of modules of interest, right. What are the modules that are interesting to us? So, when we do the network generation, we will see a lot of modules that are there, but then we will identify some modules that show a different pattern between cancer and normal samples. Okay. Those are the modules that are of interest to us. So, we will identify these modules, and then we will analyze expression of some of the genes that are in there in those modules. Okay. So, based on these, we can perhaps then identify most interesting targets, etc. Okay. So, we will do this in the hands on, right. So, we will first do the soft threshold, and then we will do the network construction, where we will identify modules using the soft threshold. Okay. So, this is the agenda for this class. So, we will do the soft threshold determination followed by network construction. Okay. So, let us now move into the hands-on part in R. So, here we are, right. I am showing you, taking you back to the data set, right. We did a lot of pre-processing, normalization, etc. on this data. So, I will just load that. Okay. So, just to remind you again, the pipeline, right. This is the data we have loaded using the reader package from the IDVerse library. So, this is the data with diffuse gastric normal raw count data. We did the drop NA value first. This is something what we did. Then we did the normalization, data normalization, as I mentioned just now, and followed by that we did variance stabilization, which allowed us to do gene filtering. Okay. So, this is, I believe, with which we did in the last class, and then we did the plotting, and using this plot RLE function just to visualize the expression pattern before and after normalization. So, if you remember, we saw quite a bit of difference before and after normalization. So, once this is done, now we will go into the WGCNA analysis, the heart of the WGCNA analysis, okay, which is the soft threshold determination. Okay. So, just to remind you again the terms that we used, right. So, we have this norm data, right. So, norm data is the variance stabilized data, but for only the genes, right, that show top 10% of the variance value. Okay. So, this is what norm data is. Okay. So, maybe we can have a look at that norm data first. Okay. So, what we can do, I can look at the dimension of the full data, right, where we have 39,000 genes. Here we have 3,900 genes, right. So, you can see the row numbers. So, we have taken the top 10% genes, okay, top 10% according to their variance. Okay, so, we have done that. Now, we can go into the determining the soft threshold. Okay, the function that we will use is called peak soft threshold function, and this is from the WGCNA package. Okay, so, I believe again we have seen this manual of this package, and we have seen this function in the manual, etc. We are going to use this. So, let us first load the library. Okay. Okay. So, in my system the library is installed. So, you can install it by simply saying install.packages, right. So, that is the standard way of installing. Now, the package now is attached. So, what we have to do now is we have to use this command. So, what does this command do? I will show you in a moment. Let us look at the head norm data, right. This is how the data looks like. For WGCNA analysis, what we have to do first, transpose the data, right. So, transpose the data means we have to, the columns would be the rows and the rows would be the columns. So, we will have to interchange the rows and the columns. So, that is what the transpose do, okay. And the transpose command in R is very simple, is just T, okay, T, okay, and you see this has been transposed now, okay. So, the gene names are in columns and the sample names are in rows, okay. So, we can store this in a variable called data. So, T means transpose again, right, and we can check how the input data looks like. Okay. This is how the input data looks like, right. As I said, sample names are in the rows and the gene names are in the columns. Okay. So, for many analysis in R, you have to transform the data to actually suit the pattern, right, by which the analysis can be done. Okay, so, now what we have to do, we have to determine this soft threshold, right, and we have to pick these powers, right. So, as I said, this is the power that we have to determine where we get a scale free network. For determining this power, we can determine in different ways. For example, we can sample, right, powers sequence from 1 to 19 at step of 2, right. So, if we use this command, it can do 1, 3, 5, 7, 9. Okay. So, this is slightly faster. We can also do powers 1 to 20. Okay. So, when you say C 1 to 20, it's like it will do 1, 2, 3, 4 and so on up to 20. Okay. So, this is something possible, and this is probably ideal, but then of course, it will take more time, right, to run. What we can also do, we can use power C 1 to 10 and then sequence 12 to 30. So, it will first run 1, 2, 3, 4 up to 10, then from there it will do 12, 14, 16, 18, so on. Okay. You can try out all of this, right, but for the sake of the class, right. So, we do not have a lot of time. So, this is what we are going to try out. So, we have defined the powers now. So, we can check what are the powers, right. You can see the numbers. So, up to 10 it is from 1 to 10, then it is 12, 14. So, step of 2. Okay. Now, what we will do is we will use this function, right, where we will have the soft threshold is the peak soft threshold. This is the function, okay, and we have to give the input data and the parameter that we need to specify, and this is something you will see in the manual. Power vector is equals to powers. Okay. So, we have to specify, we have to give input data and then we have to give the power vector parameter. Okay. So, in the manual you can see when you look at the function for peak soft threshold, you will see these parameters that we need to specify. Okay. So, it will take a bit of time to run. So, what it's doing, it's actually evaluating, right, how well the network that is derived is actually fitting the scale free network. Okay. So, for each of these power values, whether the network fills the scale free topology, okay, whether it becomes a scale free network. So, what you see here now, this is done, right. So, what you get is you can also run this in parallel, right, if you have parallel processing in multi-threading, which we are not making this too complex right now. Okay. So, here are the results. What you see here is the power, right. You have 1, 2, 3, 4, etc., which we specify, right. You can of course specify anything that you want like, and you can modify this. So, you might ask, why are we running till 30? We can stop at 20. Yes, of course, but again, if you run at let us say up to 20, and you see for your data whether you reach kind of like a saturation or not. Okay. And this is the SFT R square, means how well you are fitting the scale free topology, right. So, SFT R square value, the slope value, the truncated R square, mean k, median k, etc. Okay. So, we have determined. So, what we can do, we can simply type software. Okay. You will see the same thing, but there is one difference. You will see here, the first thing it will tell you power estimate. Okay. It says like probably the ideal one is 18. Okay. So, this is a kind of a suggestion from this analysis, okay, that maybe 18 is probably the most suitable one. We will see whether 18 is the most suitable one. Maybe we can do some plotting, and then we will identify which one is the best power for us. Okay. So, let us see, right. So, how we can estimate extract this. So, we can say, for example, there are two values that we can extract from here using this dollar, right. So, there is one power estimate, the other is fit indices. Okay. So, power estimate, we have seen it suggesting 18, and we can say fit indices, right. For fit indices, we can see this power, SFT R square, slope, etc., etc. Okay. Now, what we will do is we will see like whether 18 is the best one by doing some plotting here. Okay. So, what we will do is we will plot power versus SFT R square first, and then we will plot the power versus. So, there are two plots we will do. One is power versus SFT R square, and then another one is the mean k. Okay. So, here is the mean k, which is the size of the network, right. So, mean k. Okay. So, let us do that. Okay. Size of the network or the degree of the network, right. So, k that we determine. Okay. So, let us first plot this the power versus the SFT R square. Okay. And for that we are going to use this, okay, fit indices. We are going to use this here, SFT R square. So, what we are doing is, you have probably seen this function already, right. This is actually, we want two plots side by side. So, that is what we have first mentioned the layout here. Sorry. Okay. So, we will plot these two side by side. So, the first one is, as I said, we are going to plot okay. Okay. So, what we are plotting here, you can see this a very basic plot, which we can improve of course, is the power value, and then here you have the slope, right, the sine of that, right, sine of that slope times the SFT R square. What is the sine of this? You can see this here, if we just type okay. So, here the slope values, and then we have the soft threshold fit indices SFT R square. Okay. So, from here, right, we are multiplying with the slope, and then we are getting the values. Okay. So, here we have starting from let us say minus 0.4, and then we have slowly increasing to 1. Okay. So, 1 means perfect fit, of course, we are not going to get 1. Okay. So, of course, we can get a bit more nicer plot, right, and for doing that we will use this. So, this is what we are plotting, right, indices power versus the multiplication of the sine of the slope and the SFT R square value. X lab, right, this is something we already know, right, the X label and the Y label, what should it be, PCH 16, the point type, the color of these points and the main title. Okay. And then we have the size of the axis, size of the label, etc. Okay. So, let us see. Okay. So, what we have to do again. Okay. All right. So, we have these values, right, and what you can do is it seems like it's going up a bit and then it's slightly saturating here and then going up again, right, and then finally it's saturating somewhere here, right. So, maybe we can think about a threshold or a power, right, somewhere here, right. So, a power that we would like, okay, which is kind of going towards scale free topology. Okay, the suggestion was 18, right. So, 18 is probably here and then we can see whether it's what are the numbers. So, for that we can actually use this command which will put these numbers on top of these points. Okay. So, that's what we are going to do, right. We are going to use this command text, right. The x and y values, right, x values will remain the same. The y values we are using this minus sign, etc., right, multiplication of that, and then minus 0.03, right. So, we want these labels just below the points, right, slightly below the points. Okay. So, that is what we are going to do, and that is why we are going to use this command text, right, x and y minus 0.3, and then we are using the labels, right. So, labels is powers, right. So, that this power number will be given there and the color would be this dodger blue one. Okay. So, you can specify any color you like, but we said okay, we will use this color. So, this is what you see, maybe it is not very clear. So, you see this minus. So, this is the first one, power 1, this is power 2, this is power 3 and so on, the numbers are increasing. The suggestion was 18. So, 18 was just here. So, what we think, maybe probably slightly higher would be better, maybe 20 or 22, something like that. So, depending on that we can choose, right. So, maybe you can say 20 or 22. Okay. The other thing we can look at is as I said, the soft threshold versus the mean k value. Okay. So, that is the degree. So, before that we can also add a line, right. So, we can say we are reaching around 0.8, 0.9, 0.95 maybe, right. So, we can add a horizontal line that will help us kind of determining where we are kind of reaching saturation. Okay. And then we have this AB line H equals to 0.95 and LTY equals to 2. Okay. So, line type 2 at value of 0.95 we want to add a line. Okay. And here is the line, you can see, right, that kind of helps us picking the right threshold. So, we can pick around maybe 22 or 24, right, we are reaching this kind of level of saturation. Okay. So, we will pick that, but the other thing we also want to plot is the, as I said, the power versus the mean k value. Okay. We use the same command, the plot xy, right. This is again plot xy. These commands are already familiar to you. We are using x labels, y labels, the point type, color, which color we want, and the main title, and the Cx axis and the Cx dot label, right. So, these are the all of these are familiar, right. They should be familiar, we have, because we have used them now many times starting from the initial class, right, for visualization. Okay. So, this is how it looks like. So, as you can see, like beyond certain soft threshold, the mean degree kind of saturates, right, there is not so much change anymore. And as before, we can also put the numbers on top of these points, right. So, the x and y value, x value remains the same, y value we just putting plus 50, so that the labels are above the points, right. So, the labels appear above the points. Okay, you can see this here. So, labels appear above the points. Maybe these are not very clear, but if we make this plot bigger, you can now see these points here. Okay. So, from these two plots maybe we can pick around 20, 22, right. The suggestion was 18, maybe I will go with 22 or 24, right. So, anything we like, that is okay. So, we can say okay 22, okay, maybe that is the right number. Okay. So, based on that we first define this number. So, chosen power is 22, okay. But again we can try out different numbers, the network modules will be different of course, but you will get pretty similar results with some minor differences. Okay, so, we have chosen this 22 as our threshold, right, that is the soft thresholding part. Okay. So, now we have done the soft thresholding part, what is remaining is now the network construction. Okay. And for the network construction we have to use this command blockwise modules. Okay. This is again part of the WGCNA package, and I believe last time we have seen this the blockwise modules package. Again we have to give the input data, we have to give the power, right, which power. So, this is the power that we have chosen from our analysis from the soft thresholding, right, and the network type that we want. There are several other parameters that I have just kept. You can again go through the manual and see what do they mean actually. So, this is what is the mean module size, max block size, right, how big a block can be, right, how much is allowed, etc. Whether we want to save these TOM files, I will come to them a bit later in the next class, what are these TOM files. So, that here it means true. So, what should be the save TOM file base, gastric cancer, that should be the name of the output file, output TOM files, etc. Numeric labels is equal to true. So, we can actually see what all these means. Okay. Otherwise, if we do not want to touch them first, we can run this with default parameter values, whatever they are, we are just saying the power and the input data, but then we can slowly start optimizing and see what they mean. Okay. So, this is we have this option. Now, we have determined this power, chosen power, right. So, we can see that the plotting actually allowed us to identify the correct power value, right. The suggestion was 18, but we actually thought maybe 18 is not the best one. So, we have chosen 22. Okay. So, before we go there, there is a correlation function that WGCNA uses, right. So, what we are doing is, because it will call correlation, we want to there is a default correlation function in R. So, we want to store it and replace that the default correlation function with WGCNA correlation function. Okay. So, this is what we are doing in the first step. So, we are storing the default correlation function in this temporary correlation, and then we have applied this WGCNA correlation function to this correlation function, right. So, when WGCNA calls correlation function, it will call this correlation function. Okay. So, once this is done, the first thing we will do is the blockwise modules. Okay. So, this is the network blockwise modules, input data where we give the power, and the network type means signed, we want signed network, and with some of these parameters. Okay. Okay. So, again it will take a bit of time, right. This is running the this blockwise modules, it is running the identifying the network. So, it might take a bit of time. So, in the meantime, right, what we can do is we can actually now look into this, right. So, once we have this network, the next thing we are going to do is we are going to look into the network, and then we are going to visualize, right, and identify the interesting modules. At the same time, we are going to save these modules that we get, right, and then we will use this data for other analysis. Okay. So, it seems it is done now, right, this analysis is done. What we can do is we can, so here we are restoring this back, right, the correlation function that we stored in temporary correlation, right. So, that is done. Now, let us look at the summary of the network. Okay. Okay. So, here we have summary of the network. So, what we are getting is something like this, right. So, we have block genes 2, TOM files 2, okay, and dendrograms 2. So, which means that we have two sets of these dendrograms and TOM files, okay, topological overlap map. Okay. So, we are going to look into them just briefly, but let us first store these figures, right, save these figures, right. So, sorry, the file name is okay. So, we are saving this file using this command dev.copy to PDF, the file name and the usual parameter, so that the quality is maintained, and then dev.off. Okay. So, this file is saved now. Okay. So, now we have seen the network, right, summary of the network. We have seen there are these 2 TOM files and 2 block genes, right. So, we will have these 2 blocks which will visualize in a moment. Okay. So, first what we are going to do is use a very interesting command that is available in WGCNA, and it is called labels to colors. Okay. So, what it will do, it will define this colors, right. So, from labels to colors, right. So, the original network colors, right. So, you can see this 114 etc. So, what this 114 etc. means is that these are part of different modules. Okay. And what we are doing is, by these colors, labels to colors means it will convert these 0, 1 to 14 etc. into different colors. Okay. So, we can see the colors now. So, that when you visualize, these different modules will appear in different colors. Okay. So, that is what this function will do, labels to colors. Okay. Okay. Again, let us go back, right, to summary of network, right. You can see this M is 18 means we have actually 18 modules, right. So, we have to have 18 colors. So, we will have 18 colors in there. Now, what we are doing is we are actually plotting this for the network dendrograms. Okay. So, we can see what are the parameters that are available to us here. So, you can see colors, good samples, TOM files, M is okay, etc., good genes, block genes, etc. You can explore this. For example, we can, as we said, we have seen colors already. Let us say we have it's running a bit. Okay. So, it's maybe it's something that we cannot make sense of that easily, right. Okay. And here you are seeing, right, this is the original data. It looks slightly different, right, the original normalized data that we actually took this for analysis. The rows are the sample names again, but the columns, right, they look different. These are not gene names anymore. These are actually saying which modules these gene belong to, right. So, ME9, module 9, ME2, module 2, etc. right. So, instead of genes, now we have this module association for every gene, and there are 18 modules that have been identified by this analysis. Okay. So, that is what we are going to do is we are going to see this the one of these dendrograms, right. As you can see, these are in different colors, right. These modules are in different colors. As you can see, you have green, blue, etc., and this is the relationship between these samples. Okay. We can also do this for the second one, right, and this will be different, and as you can see, the colors are different, right. So, it has been stored in 2 of them, right, 2 files. So, if you go back into the summary file, you can see, right. So, block genes equal to 2, right, and TOM files 2. So, we have 2 such module files. Okay. So, now what we can do is we can now store these modules, right. Modules we can save them as data frame, genes equals to names of network colors, and colors would be labels to colors network colors. Okay, so we have already defined colors anyway. So we are creating a data frame, right, where we are using the names of these variables, right. So you can check what are the names here. Yeah, so these are the gene names, right, and then we know their colors. So colors means this is the association with a specific model, right. So genes that have same color means they are head models here. So this is what you get, right. We have created a data frame where we have genes, right, in one column, and then we have colors in the other column where we are associating genes with specific colors, right. So two genes having same color means they are part of the same module, right. So you can see like two genes here, for example, in brown, and both have brown color means they are part of the same module. Similarly, we have two genes that are part of the same color here, turquoise, so they are also part of the same module. Okay, so we have defined this module. What we are going to do last is the save them, right. So we have do the write table and we have saved that in file, okay, WGCNA modules.txt. We can load this data, right. We can simply load this and see the data. Okay. Okay, genes and colors. So this is now stored in that file. So we can remove this from the environment because this is not required. Okay, so we have done the soft thresholding. We have determined the power, right, the threshold that we are going to use to get to the soft scale free topology, and then using that threshold we then constructed the network. We have seen different modules and we have seen some of the visualization, and we have also stored the data, right, with the file within a file, right, where we have associated genes with specific colors or modules. Okay, so with that we move on to the presentation, right. So what we have done in this class is we have taken the normalized data and then we have determined the soft threshold for getting to scale free network topology. And when we determine the soft threshold, we then use that threshold, the power threshold, to actually identify modules using the WGCNA package. So what we are going to do next is we are going to identify the interesting modules and look at their gene expression patterns, the genes that are part of those modules. So that will be in the next class. Thank you.